This presentation, we are going to discuss about the Koch postulate. So, what do you mean by a Koch postulate? A postulate means a statement that is believed or accepted to be true. So, in this, the outline, what I'm going to cover is introduction, principles of Koch postulate, organisms identified by Koch postulate, and the limitations of Koch postulate. So, Koch postulate was developed in the year 1884 by Robert Koch and Frederick Loeffler. And this was published in the year 1890 by Robert Koch after making some modifications after their own findings. It is a set of criteria that aims to establish a relationship between the causative microorganism and the disease state. It provided a great tool for understanding the disease state in the early 19th century. But further understanding of the disease state has made it very clear that Koch postulate it simply points to the importance of rigorous research before making the conclusion of the causative organism of the disease state. Principles of Koch postulate. So there are majorly four criteria which is designed by the Koch and before establishing the causal relationship between the microbes and the disease condition. These are the postulates. Number one, the microorganism must be found in abundance in all the organisms suffering from the disease, but should not be found in the healthy organisms or maybe the host. The microorganisms must be isolated from the diseased organisms and grown in pure culture. The cultured microorganism must cause a disease when it is introduced into a healthy host or organisms. And fourth criteria, the microorganism must be re-isolated from the inoculated, diseased experimental host and identified as being identical to the original or specific causative agent. And this is what the complete Koch postulate. The image one, it represents the diseased host. It is isolated, the infectious microorganism, and it is isolated by the pure culture. And this isolated colony or the culture microbes when it is introduced into a healthy host or a healthy organism, and that is getting disease. And later, and from the diseased host or organism, we can isolate the same and exact identical microorganism. And this was Robert Koch postulate. It's a major milestone in the disease or pathology identification. And the same when you tried with a healthy individual and the microorganism, it may not be present. And when you go for a pure culture isolation, nothing was found in the healthy host. So this was the Koch postulate majorly. The four criteria was established by Robert Koch, was published in the year 1884. And based on this Robert Koch postulate, so many diseases were identified in different years. Tuberculosis was confirmed by Mycobacterium tuberculosis in the year 1882, and anthrax, where Bacillus anthracis was confirmed in the year 1876, and cholera, Vibrio cholerae was confirmed in the year 1884. Typhoid, Salmonella typhi was confirmed in the year 1884, and bubonic plague, Yersinia pestis, it was confirmed in the year same 1894. Tetanus was confirmed by Clostridium tetani in 1889. Diphtheria was confirmed in the year 1890 with the Corynebacterium diphtheria. And even though there is a postulate with given a perfect criteria, this Koch postulate also having some limitation. The first one, at the time of this postulate was introduced, viruses could not be seen or it may not be identified in the status as per the postulates. And further studies of the disease of pathogenesis and pathophysiology showed that an organisms may be the causal microbes, but it may fail one or all four criteria of the Koch postulate. So this is illustrated with the following conditions. Number one, some pathogenic organisms are found in the healthy organisms, but it produces only disease to a few individuals who are infected. The examples are Bartonella species or herpes simplex virus type 2. And limitation number two, the viruses and other pathogens don't grow in the pure culture medium, and they were used to the term HACCP. A HACCP, it represents, the H represents Haemophilus and Haemophilus para-influenzae, and A represents the aggregative bacter, and including aggregative bacter actinomyceto Mycetome comitans, and Aprophilus. And the C, it represents Cardiobacterium hominis, and E, it represents Echinilla carodens, and K, it represents Kengerilla, or particularly Kengerilla kingae. And limitation number 3, the complications of the prion. So prion is nothing but an infectious or abnormally folded protein. So these are the non-living organisms that cause disease, but these diseases require specific physicochemical conditions as well as the host susceptibility before it causes a disease. So Koch postulate expects pathogen to cause a disease in all healthy organisms, but this is not the case with the case in the case of prions. And many lesions as well as cancers, which is caused by the pathogens or without the pathogen causing a disease. And this is found in some cases of rheumatic fever caused by a group A streptococcus infection and cancer due to papillomavirus infection. And various modern approaches in pathogenesis of today's scenario. So that helps in the identification of many pathogens as well as in organisms. See, modern approach to the pathogenesis of infectious disease have shown that the diseases are usually due to the multiple factors coming together, and the opportunistic infection so clearly speaks to inability or pathogens to cause disease in an organism while it does it in others. And modern approaches consider all the steps involved before the occurrence of the disease state. The ease of genotyping species of organisms has made it very easier nowadays. And Koch postulate may not be satisfied, and disease conditions still attributed to a specific pathogen. So, and finally, this Koch postulate, it concludes or given an idea, a rigorous study of the disease and their pathogenesis and major disease conditions were understood as a result of Koch postulate. And further research in pathogenesis of disease and the nature of some rare disease conditions has caused for re-evaluating the procedure of establishing causal relationship between the microbes and disease. Thank you.